welcome Dr. Komal Kampasi here. So she will be giving her talk on. So Komal is a strong advocate for neuroscience and optical neurotechnology lead at Lawrence Livermore National Laboratory at California, where she also serves as USA Federal Research Wings of Science and Technology. She received her doctorate in biomedical engineering from University of Michigan. Her research interest includes brain machine interference, nanotechnology, and medicinal devices. She is also part of various neuroscience outreach initiative, which focuses on creating neuroscience and mental health awareness at grassroots levels, especially for the young and the underserved communities. So I welcome you, Dr. Komal. Over to you.
Thank you, Dhanashree, for the introduction. Let me just switch over to the slides and share my screen and we can get started.
Yeah.
Okay, perfect. And if I do face any connection issues, I might switch off my video for a while. So hope that's okay. All right, thank you for having me here today. As introduced, I'm a research staff at Lawrence Livermore National Lab in California. And in my today's talk, I'm going to talk about the technique of optogenetics and the recent advances and future direction. This is one of the revolutionary techniques in brain science, and hopefully we can get some interest generated among the audience to know more about it and pursue the field if they may. This is also one of the actively researched field in our lab right now, and I'll talk a little bit more about it. So Lawrence Livermore National Lab is a federal research facility situated in Bay Area, California, and is one of the 17 DOE labs, which are Department of Energy labs in the country, and it was founded in 1952. Here in the Bay Area, it's situated, the main site is at Livermore, and it's about a 1 square mile campus with all facilities on site. Towards the east of it, we also have an experimental test site, which served as a nuclear test site during World War II and has been active since then. And recently we had a campus opened in San Francisco to keep collaborative ties with companies and startups in the Silicon Valley. The annual federal budget of the lab is about $2 billion US, and we are a nonprofit, and a lot of that funding comes from the US Department of Energy. And there is a highly qualified technical workforce that we have on site, including postdocs, undergraduates, contractual employees, staff, scientists. And within at the lab, we have different divisions. Within the science and technology division is where our groups called implantables neural engineering group is positioned, and that's where I and my team members conduct the brain research. So having said that, in today's talk, I'll now start with a brief introduction to what human brain is and neurons are and what neural interfaces are. I'll be primarily focusing on invasive brain implants for this part of the talk. I'll then talk about optogenetics and introduce some advanced tools which are used to do optogenetics in animal and human models. And then I'll conclude the talk with discussion on the future of optogenetics, both in research and clinical applications. So to understand what neural interfaces really are and how they work, first, let's take a look into human brain, which is a supercomputer inside our body. And it may not look anything more than this pinkish grayish flesh from outside, but last 100 years of neuroscience have allowed us to zoom deep into the structure and see that it's actually made up of hundreds of billions of neurons, which come in these different kind of shapes, size, and are made up of different molecules. They connect with each other and form these connections to pass on information. If you zoom in into this one such connection, let's say there is information that needs to be passed from this neuron A to neuron B. Neuron, by the way, is a brain cell. And how that works is that neuron A transmits a neurotransmitter, which is a chemical. And once neuron B takes that chemical in, the action potential, the membrane potential of the cell, it goes from the resting minus 70 millivolt to about 40 millivolts. And there's a huge surge in the potential of the membrane, which is what we call as an action potential or spike in neuroscience. And by communicating, by passing on this action potential or neural spike from one neuron to the other, this is how the information gets communicated inside the brain. Now, if there's any brain area that's like damaged because of, let's say, any neurological disorder or any injury, what happens is that these connections, they get hampered, and either the excitability between the neurons, like there's more spiking than usual or less spiking than usual, and that becomes a concern. So what neural interfaces are, they are like any interface or any kind of a device or a chip that can mimic the function of these damaged junctions by generating electrical pulses where and when needed, is what you call a neural interface. So that's what neural interfaces are. Now, talking about these interfaces, I feel that we as humans, we have long been fascinated by brain implants for a long time, from science fiction shown in Hollywood movies, where you see things like intelligent machines in Matrix, to life-size human beings, robots in Transformers, to even like a neural handshake shown in Pacific Rim, where two minds of two different people could sync with each other. So it's just that now these interfaces are now turning into an actual reality. And the next slide shows how the real world commercial interfaces look as per date, which is pretty close and very cool. The first of its kind, here are a few of the interfaces that have been out over the last few decades. The first of its kind was released on a commercial platform, was by a company called Cochlear in 1981, which was a cochlear implant, which is essentially an implant for your ear for people with hearing disability. And it could help people with hearing disability hear again. The next is a deep brain stimulation device that was introduced in 1997 by a company called Neuropace, and is actually currently making a lot of buzz because of its improved efficacy in in treating patients who are suffering from epileptic seizures. And how this implant works is, again, it's implanted invasively inside the brain, and the electronics and the power gets managed on a extension of a device that's just plugged onto the chest of the patient. The most recent to these interfaces that with FDA approval are retinal peripheral interfaces. One of them is a retinal interface as shown here, which is, it interfaces with the neurons in the retina, which is at the back of an eye, and helps blind patients see again. The implant shown here is actually a retinal implant, which was the first FDA approved implant in the USA, and was actually made by the implantables group at Lawrence Livermore Lab, and then licensed by a company called Second Sight. So all of these interfaces that you see here are mostly electrical, which is what it means is that they interface with the neurons in the brain by using electricity. So they generate electricity, and they make the neurons, they put that electricity into the brain, and that's how they work. But now, instead of electricity used in these implants, what if we could use light? And let me explain that how. So let's again zoom into the segment of the brain again, and say you have this particular kind of cell that needs to be manipulated, that needs to be fired, or that needs to be fired into an action potential. So what you do is that you first genetically modify it so that it becomes responsive to a certain wavelength of light. And this genetic modification is done by introducing a protein which is sensitive to a light wavelength in a viral vector and inject it into the brain. So when now you shine light of that particular specific wavelength in that brain region, just these cells are going to be driven, while rest of the cells, they remain isolated. Means the cells in the background, they would not change their working, they would keep working as they would. So similarly now, after, let's say you want to manipulate the second group of cells at the back, let's say cell B type, and you can genetically engineer them to be responsive to another wavelength of light, let's say red, and so on. So by genetically encoding different types of cells with different kind of protein, you could actually control different cell populations in the brain with different colors, which is pretty cool, and something that could not have been achieved ever with electrical stimulation before, because you don't get this kind of selectivity and specificity to control different neural groups with different light techniques. So this field of brain research is basically what's called as optogenetics, as it uses light to manipulate genetically encoded cells in the brain. And then there are advanced tools that allow you to do so, and those are called neural optoelectrodes, and that's what I'm going to be talking about next. So here are some of the recent state-of-art invasive optogenetic tools. One of the primitive ways that was introduced like about 2 decades ago, when the technique actually came into being, was delivering light into the brain using an optical fiber, as you see in the picture. Here, a thick optical fiber was kind of stuck into the brain of the animal, and this technique, that's why it's like highly invasive, because these fibers are pretty bulky and pretty big. And even though like it has its disadvantages, it's still one of the widely used techniques because of its simplicity, and also that you can very easily couple the fiber to a laser that sits on a bench top and control the power, the frequency, and so on of your stimulation techniques. The second type that came into being was a fiber coupled to a tiny waveguide on a neural chip. So let's say there's a neural probe chip that's about like hundreds of microns in dimension. This part of the chip that actually goes inside the brain and is the actual implant is about tens of microns. And there's a tiny waveguide, which is a structure that guides light into the brain, is situated on it. And it would be at the back end of this waveguide that you would have such kind of an optical fiber. So what you're essentially doing, instead of putting that fiber in, you are coupling that fiber to a tiny waveguide, and the waveguide actually goes in. So this technique is less invasive because this waveguide is just tens of microns, and is also compact because now you have a very small implant sitting on the animal head rather than a full optical fiber. And then next kind of design is where light sources are directly placed on the tip of the neural probe, like you see here in this picture. And its foremost advantage over the last couple of approaches is that it's the light sources, there are no fibers involved at all. So there would be no fibers hanging from the head of the animal. It gives you ease of experimentation. And the only kind of potential concern is that now the light source is placed inside the brain. There is a potential concern of brain heating with prolonged use of the device because of the thermal heat generated from these light sources. The next kind of technique that scientists came up with is where they would manually assemble fibers with these light sources, and then couple them to these neural probe shanks, which are the shanks that actually get implanted inside the brain. Now, since this technique used commercially available light sources, you can actually make devices which are multiple color. So you can control as many brain population or brain cell types as needed. But then again, the drawback was that it is labor intensive and not repeatable and not reliable, because you have to manually assemble parts which are like hundreds and tens of microns. So you can just think how laborious that could be. So while I was while I was in my grad school and working on my PhD, the aim of our team was to develop a technology that not only has some of these advantages of these techniques, but could integrate all of them, all these merits into a single optoelectrode design. And that's what our team achieved through completion of the fiberless multicolor optoelectrodes project, where we made this device that was a multiple color neural optoelectrode. And how we realized it is by having the series of light sources assembled at the back end of the device. These are the shanks of the device that would actually go inside in the brain and be implanted. And what was significant was this optical mixer design, where different mixer arms would actually converge into a single waveguide. So by just putting different color of light sources at different arms of the mixer, you would actually be able to get different color light at the output. And everything was done through microfabrication, so nothing was manual assembly, and hence the devices you would get would be repeatable and reliable with repeatable results. This is the device, the real prototype that our team built. And this is a device in working, where you would see different colors coming out of the shanks of the device that was actually implanted inside the animal. And here's the whole device compared to a US dime in size. And these are the 4 shanks that would be implanted in, with mixers at the back end. And then these are the light sources, which are directly coupled to the waveguides, but they are far enough from the brain to not cause heating. Validation and usability of these tools in animal models for circuit dissection was done with our collaborator lab at New York University. And I have only one slide on animal data because I wasn't sure if how interested the audience would be in it. But of course, for anyone who wants to read more about it, we have published work on this. And so feel free to Google it, and you'll be able to pull that research up. But just to explain the technique, I still had this one slide in here to kind of walk you through how this technique really works. So this slide shows the animal data that we collected from the hippocampus of mice brains using these devices. The hippocampus of these mice, they were genetically modified so that the pyramidal cells, which is a kind of cell in hippocampus, would express 2 different kind of these opsin proteins. One was that could be responsive to blue light, and the other was made responsive to red light. And both these opsins were put in the same pyramidal cell. So the data on the left that you see is actually brain activity data with voltage on y-axis and timestamp on x-axis. So when there is no light coming out of the waveguide, the brain activity is happening as usual with these random spiking events in between. But when the blue light is turned on, for that period of when the pulse is on, we saw this higher neural activity. These are neural spikes, neurons firing for precisely for that period. And there was no spiking before and after this event, which means that we were able to successfully turn the neurons on by just shining the light on in that particular brain region. And similarly, when the red light was on, a different kind of opsin was responsive to that light. And what that opsin did is that it inhibited the neural spiking. So if the spiking was happening in the brain before and after as usual, but as soon as the red light was on during that period, you could see the spikes were inhibited, or in a way you can say that you could turn the neurons off by using red light. The data on the right shows analyzed data from the same animals, where again, the results are the same, that when the blue light window was on, neuron activity was higher than usual, and you could see a lot of neurons firing. For the red period on, the neurons were not firing as much, so you could see a dip in neural spiking. And among all the neurons that were recorded with our device, if we isolated one brain cell, we saw that the same brain cell was able to turn on during the blue light period, and the same cell was able to turn off its activity during the red light period. Hence, we kind of showed this bidirectional control of a single cell, where you could, by just changing the wavelength of light, make the cell fire or not fire, which is pretty cool. And this is something that we had kind of proved for the first time using these devices in animal models. So moving on, as I joined the research group at Livermore, now the aim was to further improve the lifetime of these devices in tissue. And one of the most effective ways we could do so was to replace the semiconductor material that was used in the making of these devices, semiconductor silicon, and replace that with softer polymers, softer materials like polymers, so that you could make more flexible devices that could mechanically form a better interface with the tissue. And that's what we did with this prototype that was designed and made from all flexible substrates. You could see this like very thin film hair-like structure that had all similar components as the previous design I talked about, with a waveguide on the shank that would be implanted inside the tissue and electrodes to pick neural activity and connectors on the back end that would be sitting outside the head of the animal. So this device kind of had all the advantages, engineering advantages of the previous design, but at the same time was also made of all flexible materials to increase the lifetime of the device and increase the longevity of the device in animals. So in this slide, what I'll show you is a quick demo that summarizes how these devices are actually made, and it should give you a generic idea of how neural interfaces are generally designed and assembled in this space. We term this as POEMs, this interface, because it's polymer optoelectronic device. So we start with a silicon substrate, put a thin polymer in, define metal sites, which are to record neural signals. Then we insulate with more flexible films, pattern our waveguides, then put more waveguide insulating films on top, expose the electrical contact so that it can be in direct contact with the neurons. This is what is replicated on the whole wafer during silicon microfabrication. And at the back end, we define again these electrical pads, which are for the laser diodes to sit in later on. And when the whole device is released from the nanofabrication facility, we put the electrical and the optical connectors on in, which would help the device to communicate with the computer and communicate with the outside world. And then at the end, we assemble these light sources on the device, which could then be electrically connected to turn on these multiple wavelengths at the device. And everything is like super flexible, unlike the previous design for better lifetime in tissue. And to insert it in, we just put it on a rigid backend to insert into the head of the animal, take the backend out, and then you only have this very thin film suspended in the brain to cause minimum tissue damage. And then by lighting different cells, depending on the requirement of your experiment, you can turn on different wavelengths to turn on or off different cells and do your neuroscience experiments. So this is how the whole device looks at the head of the animal. And if you compare this to the device I showed in the beginning of that optical fiber stuck into the head of the animal 2 decades ago, this is how the size of the implant compares. And you can see that how there is about like a couple of orders of magnitude of reduction in size and volume to give scientists ease of experiments. Again, there is like more results and more data published, so you can look that up if anyone is interested, or I'm happy to answer questions too. So this brings me to my last topic, which is the translation of optogenetics in humans, which has been debated on for a while now. Initial approvals were in fact recently granted to 2 studies, first to Retrosense Therapeutics at Ann Arbor in 2015, and second to another company called Genside Biologics in France in 2018. Both these are pharma companies who aim to restore vision in blind patients suffering from retinitis pigmentosa. Now, these 2 companies recently announced their phase 1 and phase 2 trials just this year. So it looks like things are going in the right direction. And these attempts, along with these rapid advancements in the field, they do demonstrate that optogenetics is already progressing towards clinical applications. Although approval for the first clinical trials involving these interventions on human subjects has already stimulated discussions of most of the basic dilemmas, such as efficacy and safety concerns, and a more focused debate on the ramifications of these specific interventions is still lacking. Also, as such interventions are becoming more and more plausible with given pace of research, it is crucial that we begin to address the ethical challenges that optogenetics may entail. And a neuroethical committee must start to consider the ethical challenges associated with the vast capabilities of these fields, ranging from be it gene modification in humans to long-term side effects of doing genetic engineering, to harnessing the power of optogenetics for techniques like memory enhancement and modification studies, and what the side effects of might that entail. So in summary, I would like to say that yes, it will probably take some years before scientists find solutions to current technical and safety problems of optogenetics. This is in fact true for any other new engineering technology that comes in the field. Optogenetics has proven to be a revolutionary tool for research, brain research, and we have yet to discover a lot in that space. But yes, when we talk about using optogenetics for clinical treatments in humans, some time is likely to pass before this technique, or let's say a similar technique, is approved as a form of treatment for a broader spectrum of brain disorders. So I would say that at this stage, it is also the role of neuroethicists to anticipate the more universal ethical challenges that optogenetics would eventually have to face. With that, I acknowledge my entire team of researchers who I have worked with over past 1 decade in the field of optogenetics, and also the funding agencies that have helped to fund the work that I showed here. And with that, I thank you all for listening to me, and also thank Project Encephalon for doing a wonderful work of this outreach, and also for having me here. I open the floor to any questions that you may have.